a microphone that's portable. So we're starting the next unit, Unit 2, and I looked on Blackboard this morning. I'll try to rearrange it because I like to try to put the newest unit we're working on at the top, so you don't have to always keep scrolling down. If you haven't already, the Unit 1 exam, quiz, whatever you want to call it, assessment's due tonight. But we're starting our next unit. It's 2 PowerPoints again, and it'll take us 3 lectures. So between today, next Monday, which I've got recorded, and next Wednesday. So everything in the first unit is just kind of some basics. What are cells? What are features they have? How do we look at them? This next unit is talking about what do the cells need to grow. So we've got it broken down into 2 chapters. I think yours says chapter 6 is chapter 7. They did an update, and I forgot to get the number changed. But this chapter is all specifically what do the cells need to grow. And then for our labs, upstairs, we are growing the bacteria. So like, what do we have to make sure they have so that they can grow? So what are different things that bacteria require to grow? That's all microbial nutrition and growth. The next chapter, which I've got part of it recorded and we'll finish it next Wednesday, is once they've got all their nutrients truly inside of them, what's going on that allows them to grow and divide? That's getting into metabolism. And A&P-wise, I know you guys go into cellular respiration and metabolism as well. And so a lot of it's going to be like, oh yeah, I've heard of all this, but we do talk about the differences because a prokaryotic cell is going to do some metabolism a little bit different and cellular respiration a little bit different because in your cells, cellular respiration happens on the mitochondria. But do bacteria have a mitochondria? No. But they still have to make ATP and they still have to do all that as well. And so there are some slight differences. So today we're going to get through, I've got an endpoint on where I thought we'd get to, and then I recorded the rest of my office yesterday. So that's where I want to get to today. So fingers crossed I don't have to record like a little mini lecture. And if we finish early, we finish early. There we go. So this PowerPoint talks about what is microbial growth, just what is it? What are some of the growth requirements that are needed? So we'll talk about different types of oxygen requirements, other type of nutrients they need. What are some type of physical requirements we have to make sure they have, like temperature and pH? How can we grow organisms? Because we grow organisms in our lab. And so what are different methods we can grow them? How can we measure the population growth? How can we, kind of like tracking their growth, and how do we measure that they've actually grown? I'm pretty sure it's somewhere in this area that I've got recorded. I'm afraid with having the microphone in that I can't also have my clicker. It's 2 things in the USB, so I'm just going to have to keep going over there to click forward. So first, growth requirements. What is microbial growth? So generally when you hear about things growing, you think visually in size they grow. But when we're talking about microbes don't grow in size. They're not like us where little babies become a big adult. Microbial growth is talking about the doubling of the bacteria. So microbial growth is microbes doubling in population. So it's ultimately population growth. Instead of having 1 bacteria, now you've got 1 million bacteria. And in our lab, we want bacteria to grow. Because what they look like once they've grown enough, we made them happy enough, they grew enough, is they will show up as a colony on a plate. And again, this is where lecture and lab keep going back and forth because we defined a colony last week. A colony is where one bacteria, it's a spot on a plate where 1 bacteria landed and reproduced. And that 1 bacteria became 1 million bacteria, 1 billion bacteria, and it's now visual. We can actually see the bacteria cells because there's so many of them. Not because the cell grew, it's just there's so many of them, they're now visual. So it's spot on a plate, 1 bacteria landed, and it multiplied. In your lab books, you have this exact same picture. We use this, and we'll start using this next Wednesday once you grow some bacteria out Monday, is I said, last week, you're gonna have to describe what isolated colonies look like, and there's lots of different ways to describe what isolated colonies look like. I always want to know size. I always want to know what color they are. I don't care about the word pigmented. I always want to know what color they are. But then there's lots of other ways to describe what colonies, what that microbial growth looks like on a plate. I'm like, is it completely transparent? Is it not? Like it's like white color, you can't see through it. Is the texture smooth or rough? Is it shiny? Is it kind of dull and flat? If you look at that margin, is it like a perfect circle, or do you see kind of some ridges? Some might look more filamentous. There's different shapes. There's different elevations. You know, there's lots of different ways we can describe what colonies look like, and different bacteria species do have very distinct colony morphology. And so again, our first clue on what a bacteria is always what does it look like on our plates. Now, growth requirements, things that organisms need to grow, bacteria specifically. They need nutrients. So they need elements right off the periodic table, just like we do too. And they need energy to be able to put everything together. So they need basic nutrients and they need energy to put it together to make whatever molecules they need. Now, the 4 basic elements that microbes need for survival, and these ones I all mention and we talk about, carbon, hydrogen, oxygen, and nitrogen. Spelled Chan, which is not a real word, but carbon, hydrogen, oxygen, and nitrogen. All bacteria need some requirement of these different elements. And then they need some type of energy to put them together to grow. Organisms can't grow without having some type of energy source. Where they get their carbon and where they get their energy source helps us group different types of bacteria. So we can group bacteria into categories just based on where do they get their carbon and where do they get their energy. So sources of carbon. Autotrophs means this is a bacteria that can make its own carbon molecules. Means it doesn't have to ingest them. It can make its own carbon molecules. Heterotroph means they have to get their carbon from some type of food source. So 2 places they can get the carbon. 1, they can make their own carbon molecules, autotroph. Auto means self. Like if you drive an automatic car, it shifts by itself. You don't have to have a shifter. So they can make their own carbon molecules. Heterotrophs have to ingest them. Now, where they get their energy. If you are a chemotroph, it gets its energy from chemicals. And usually it's the breaking down, breaking bonds of chemicals. Any guess where a phototroph gets its energy? From light, like photosynthesis. It gets its energy from light. There are bacteria out there that do get their energy source from light. None of the ones that affect humans get the energy from that. But there are groups. Now again, as I said, where they get their carbon, where they get their energy source, we can group bacteria then into 4 different groups, and we just smoosh the words together. So if you're a photoautotroph, you get your energy from light and you can make your own carbon molecules. If you're a chemoautotroph, you get your energy from chemicals and you can make your own carbon molecules. If you're a photoheterotroph, you get your energy from light, but you still have to ingest your carbon. And then chemoheterotrophs, which is the most common group and the ones that affect humans and cause disease, so we care about them the most, they get their energy from chemicals and they have to ingest their carbon. So again, we can group organisms based on where they get their energy, where they get their carbon. We just smoosh the words together. Now, the hydrogen, I don't even give it a slide. I give it a little blurb at the bottom of this slide. Where do organisms get their hydrogen? 2 ways. If you're an organotroph or a lithotroph. An organotroph gets its hydrogen from organic molecules, and an organic molecule means it's carbon-based. So if you're an organotroph, your hydrogen molecule comes from your carbon molecules. An example, 6H12O6. What is that molecule? It's glucose. You're going to see that a lot next Wednesday when we lecture. It's an organic molecule. It's a carbon molecule. but it also has hydrogen. So an organotroph, as it's getting its carbon from an organic molecule, it's also the same source of the hydrogen. A lithotroph just means it gets its hydrogen from something that's not organic, like hydrogen sulfide. There's no carbon molecule in there whatsoever, but it still has the hydrogen molecule to it. Now, the oxygen requirement, which gets lots of slides because there's lots of different types of oxygen requirements depending on the organism. Now, organisms, I was going to say, are either end of the spectrum, and then there's a whole bunch of grayish area. A lot of organisms are what we consider as obligate aerobes. Obligate is a term for saying required. If you're an obligate aerobe, you require oxygen, or you don't live. We are obligate aerobes. We require oxygen or we don't survive. An obligate anaerobe requires 0 oxygen, which means if you put it into an aerobic or an oxygenated environment, it will not survive. So if you're an obligate aerobe and you put it into an anaerobic environment, it will die. If you're an obligate anaerobe and you put it into an oxygen environment, it will die. And part of that is because there are toxic forms of oxygen. Normally when we talk about oxygen, we're thinking O2, which is generally a harmless oxygen molecule. But in metabolism, as things are getting broken down, even breaking down glucose because it has oxygen, you can get toxic forms of oxygen. And generally a toxic form of oxygen usually means it's unstable. It's charged. It usually has a negative charge to it. And if you have a toxic form of oxygen and the bacteria can't figure out how to survive that, get rid of it, neutralize it, oxygen will kill them. So toxic form of oxygen, they're highly reactive. It's usually because they have a charge to it. They're excellent oxidizing reagents, which we go into oxidizing more next week. But it means it wants to lose electrons. And it causes damage to cells. Examples I've got on a little picture down here. If you have a single oxygen molecule, normally we talk about O2 always together because it's stable when 2 of them are together. But if it's just one single oxygen, something called a superoxide radical, which means it's got 1 negative charge, or a peroxide anion, which means it has 2 extra electrons, they're now unstable. So again, it's if you're starting to add extra electrons, making the oxygen unstable, it will kill bacteria. So most of the bacteria that affect us survive in oxygen. And toxic forms of oxygen are always produced. Even our own cells have to figure out how to deal with toxic forms of oxygen. So do bacteria. So if there's a bacteria that survives in an oxygenated environment, which majority of them do, they have to figure out ways around these toxic forms of oxygen. And usually they produce some type of enzyme. So the superoxide radical that just has 1 extra random electron, bacteria can make enzymes. Enzymes like to end in ase. For example, this bacteria can make something called the superoxide dismutase. Just an enzyme that can neutralize superoxide radicals. Peroxide, bacteria can make catalase and even peroxidase. Again, more enzymes, they like to end in ase, that can neutralize these toxic forms. If a bacteria cannot make these enzymes, oxygen will ultimately kill them. It's not instantaneous, but ultimately oxygen will kill them because they can't neutralize those toxic forms of oxygen. Which means then we again end up getting into the obligate anaerobes. An obligate anaerobe requires a 0 oxygen environment. If you put them into an oxygenated environment, these toxic forms of oxygen, these highly reactive forms of oxygen will ultimately kill them. So we do have anaerobic environments in our body that these can cause issues at, and if we want to identify these bacteria, we have to grow them in a lab in an anaerobic environment. Now, We don't always know when a patient comes in, all right, well, they've got a bacterial infection, and we can see that. But how do we know right away, well, is it an aerobic bacteria, an anaerobic bacteria, or then there's that whole grayish area somewhere in between? When we grow bacteria, and we'll do a lot of this in our lab this semester, one of the ways to know for sure that something is a strict or an obligate aerobe is that we can grow bacteria in a tube that has agar in it. At the top of the tube, because it has a loose-fitting cap so oxygen can get in, the bacteria will grow only where there's oxygen. So if it's an obligate aerobe, it's only going to grow where there's oxygen present, and nothing's going to grow at the bottom of the tube because oxygen can't make it all the way through that agar all the way to the bottom. So just looking at certain tubes. So obligate aerobe, but it will only grow where there's oxygen. I don't know why I had this swipe weird. An obligate anaerobe, you will only see bacteria growing at the very bottom of the tube where there is no oxygen, because the oxygen will kill the bacteria everywhere else in the tube. But then we have this grayish area. There's something called facultative anaerobes. They are aerobes, even though they have the word anaerobe. The word facultative means they can do both. I always like to think, it's like faculty, we can do it all. They can in an aerobic environment, but they can grow in an anaerobic environment too. Where do they prefer based on the tube? They prefer oxygen. They will grow better. They are happier in an oxygenated environment, but they can still grow in an anaerobic environment. So they're called facultatively anaerobes, so they are aerobic, but they can grow in an anaerobic environment. So we can see them growing in both areas, but we can also see that difference that more of them like to grow where there's more oxygen. And then there's some bacteria out there that are called aerotolerant. And when I have that word aero, we're talking oxygen. They're aerotolerant. These are organisms that do not use oxygen at any part of any of their metabolism. They don't use it. There are no toxic forms of it. They just survive in the environment. They just tolerate it. So it's a uniform growth. They don't care. They don't grow happily where there's lots of oxygen. They don't grow happily where there's no oxygen. They just tolerate it. If it's around, great. I don't care about you. And so we see them just growing uniformly. We don't see them anywhere in specific. Oxygen has no requirement. Hi. You found. This is Leanne. She will be your teacher on Monday. Yep. Oh, I was gonna message you after this. My E. coli grew.
Oh, I have E. coli and Staph aureus.
Okay. Did you get it from Bill? Because I came in this morning, I set up 4 plates yesterday. And I was like, it grew.
They're good streams.
Okay. Maybe I'll use those because mine are. What?
Where do you want them?
Are they all sealed up? I'm going up to lab after this. And take it so you guys have bacteria for Monday to grow. So, yeah. Yep. You'll see her Monday. And then later in July. Thanks for stopping in. So aerotolerant, again, they don't care anywhere where oxygen is in the tube. They just tolerate it. And then my last one, I don't have a picture of in it tube-wise, but they're called microaerophilic. And we'll talk about some of these in our lab as well because we grow this semester organisms that are considered microaerophilic. Now again, I like to break apart words as much as I can, and you'll see that in lab and in lecture. I also taught med-term for a long time. The word "phil" means it likes. What does micro mean? Small. What does aero mean? Oxygen specifically. So a microaerophile, any guess? It likes just a little oxygen. So what you're breathing in right now is about, I think it's like 16 to 20% oxygen. I can't remember the exact percentage. And it can grow in that, but it would prefer it if there was less. It still needs some. It can't grow in a 0 oxygen environment, but it would prefer less oxygen to grow. the happiest. And we work with some organisms this semester in our lab that are considered microaerophilic. And so we have to provide them a microaerophilic environment. I'll talk, I think I recorded it, on how we can do that, because we do that in our lab this semester. Now, the last of my CHON is the nitrogen. All microbes require nitrogen. What's nice is that there are some bacteria that can take nitrogen from the air. So I said you guys are breathing about, I think it's like around 16% oxygen. About 80% of what you guys are breathing in right now is nitrogen gas. Just don't use it for anything. And nitrogen gas that's in the air around us, we can't use it for anything. It's an inert gas. Some bacteria can't use it either. In this form, they can't use it. But some bacteria have been able to figure out, well, how can you take nitrogen gas that's in the air and turn it into a usable form that you can use it as a building material? Because there's lots of nitrogen in proteins. And so they can do a process known as nitrogen fixation, and they can turn it into other forms of nitrogen, like nitrate and nitrite and ammonia. I don't care that you know specifically what those are, but they can turn it into other more usable forms of nitrogen. Some, not all bacteria can do that, but some bacteria can. Now it's the nitrates, the nitrites, and the ammonia. Other organisms need those same nitrogen molecules as well. And because bacteria have figured out how to make them from the nitrogen around, bacteria and plants have formed some close associations with each other. Nitrates are one of the big fertilizers you add. If you ever add fertilizer, Miracle-Gro, anything like that to your garden, if you look at it, a lot of it's nitrogen-based. Plants need nitrogen. Plants cannot take nitrogen gas from the air and turn it into a usable form, but bacteria can. And so, farming world, we figured this out, that if we can actually plant bacteria in our soil next to other plants, bacteria, if they grow next to roots, it's like the bacteria can make the usable forms of nitrogen that the plants can also then use. It's like the bacteria providing the fertilizer. It's cheap and easy. It's great. Couple other elements that a lot of organisms need, but a much smaller scale. Phosphorus, sulfur, there's some trace elements, possibly calcium, sodium, things like that, that they may need for growth. And some things called growth factors. Instead of just a single element, it's some type of compound that an organism needs for survival. And we're taking multiple elements, putting them together, like glucose. But it's a compound that an organism needs. And some organisms are very specific for what trace elements or compounds they need. And when we grow things in a lab, we then have to know what those growth factors are so that we can provide them in our media. Oh, there was my atmospheric nitrogen, turning into the nitrates and the nitrites and the ammonias. And again, plants love bacteria. Now, switching to physical requirements. So instead of just looking at elements, and I do love the Big Bang Theory, so it's not uncommon to see lots of Big Bang Theory memes throughout some of my PowerPoints. But some physical requirements certain bacteria require. One of them is temperature. And different bacteria do require different temperatures. And it's partly because of what temperature does to the bacteria cell itself. If you have something too hot or even too cold, it can affect the shape of a protein. When you change the shape of a protein, it's called denaturation. And if a protein changes shape, it can't do its job anymore. So if you have something too hot or too cold, proteins start to denature. Proteins can't do their job. Microbes need proteins. They need it for structural material. They need it for enzymes. If they don't have them, they don't survive. So they can't survive in too hot or too cold. Also, temperature affects lipids, like the phospholipids that it's in their cell membrane. If you heat up a lipid, what happens to it? It melts. This means their cell membrane would melt. That's not a great cell membrane if it's now melted. If you cool a lipid, like you freeze it, what happens to it? It hardens. Like if you've ever tried to cut frozen butter, like it is hard. Well, that's not great for a cell membrane either because stuff still has to be able to get out, stuff still has to get in, and stuff still has to kind of move and shift around a little bit. And if you harden that cell membrane, it also can't function. And so it's always kind of the bacteria, depending on the bacteria, depends on what temperature is, their happy temperature. But anything too hot, too cold can affect proteins and it can affect lipids, all negatively. So bacteria all have an optimal growth temperature. That doesn't mean if you change it a degree on either side, they don't survive. They're not going to happily survive. And if you start to go farther and farther out of that optimal temperature, they may not survive. Generally, the bacteria that cause issues in us grow best at 37 degrees Celsius body temperature. That doesn't mean they won't grow, say at room temperature, but they're not as happily growing, but they'll still grow. If you put them in a refrigerator, you can see a little tiny bit of growth, because again, it's slowing the growth. They can't survive. I shouldn't say they can't survive. They can survive, but they're not doing as well. They're not growing as quickly. They probably aren't going to cause disease if they're not growing quickly. So all organisms have their own optimal temperature. Now, depending on the different types of temperature they grow best at, we can group different types of prokaryotes to psychrophiles, psychotrophs, mesophiles, thermophiles, and extreme thermophiles. Again, they all have that phile or that phil in it, which means they like something. So a psychrophile or a psychotroph. Psychro means cold. I always think you're, psycho if you like the cold. So psychro means cold. These are organisms that are going to grow best in cold environments. If you put them in a warm environment, if you put something that's on this blue that loves freezing temperatures and you put it into the human body, will it grow? Nope. So we generally don't care about them in this class. On the other end of the spectrum, thermophiles or even extreme thermophiles, they need really hot temperature, even above boiling, for them to grow. If you put these organisms in the human body, are they going to grow? No, so then we don't care about them. I mean, we care because they're living, but we don't really care. The organisms that grow best in the human body, like kind of that medium temperature, around 37 degrees, and the green on here, are called mesophile. Meso means middle or moderate. Doesn't like too hot, it doesn't like too cold, it's kind of right in the middle. We're not boiling, we're not freezing, we're just warm. And in our lab, because we only care about the organisms that like to grow in and on us, we only work with mesophiles. So that's why we have our nice incubator up there. Doesn't mean the bacteria wouldn't grow at room temperature, but they're not going to grow as well.
Is that when our bodies have a fever?
Yes. Yes. And so if you have a fever, bacteria are not going to grow as well. And so depending on how bad your fever is, by having a slight fever and be like, I'm going to quick take an ibuprofen, can actually prolong how long you're sick. And if you can withstand that slight fever, your bacteria aren't going to grow that are in you, aren't going to grow as quickly, and hopefully your immune system can get rid of them faster. If you decrease that fever, bacteria are still growing happily. But it's when else. Obviously you have a fever of like 105, like go in. But yes, that's why we have fevers, try to get them outside of that happy growth temperature. Again, this is just showing those psychrophiles, the ones that like freezing temperatures. This is Antarctica. There is an organism that loves the freezing temperature. It just happens to have a red pigment to it. And so on the snow, you can see the red pigment. It looks like bloody snow. It's just an organism that grows happily on snow. It wouldn't grow anywhere else if it was warmer, and it just happens to have a red pigment to it. Physical requirement is pH. Organisms have a happy pH as well that they grow best at. Because if you have something too acidic, it means you have a lot of hydrogen ions, or if you have something too alkaline, which means you have lots of hydroxide ions, it interferes with hydrogen bonding, which is going to cause issues when it starts to make new DNA and during replication. So if you start to make something too acidic or too alkaline, it's going to affect replication of those bacteria. Now, microbes associated with humans prefer a neutral pH, right around 7, 7.3, somewhere in there. They grow best at. But there are other organisms out there that like more extreme pH differences. So neutrophiles, again, it has that fill word in it, likes neutral pH. An acidophile likes a more acid environment, and an alkalinophile likes a more alkaline environment. Now, there are bacteria that live in our body. I mean, most of them are neutrophiles. I don't want to say they're acidophiles, but we do have alkalinophiles that live in our digestive tract. Your small intestine, especially the very first part of your small intestine, is a more alkaline environment. It is a slightly higher pH because it's pumping out lots of alkaline molecules to try to neutralize the stomach pH, the stomach acid, which means bacteria that live in that first part of the small intestine are surviving in a more higher alkaline environment, a higher pH. We do have bacteria that can survive in the stomach, but I don't want to say they're an acidophile. Although they can withstand the acid, they don't like the acid. They just have figured out a way to neutralize it. There is another physical requirement that organisms need. Bacteria need water. If they don't have water, what happens to them? They die. And we know this. Bacteria know this as well. Now, why they need water is kind of the same reasons we need water. It's the universal solvent. Everything can dissolve in it. It's the basic fluid that has everything dissolved in it, in our cells. It's also part of a lot of metabolic reactions. H2O, water, is used a lot to break things down. And if there is no H2O present, stuff can't get broken down and then rebuilt as other things. So water is part of a lot of chemical reactions. If they don't have water, they die. But some cells still survive in a dry environment. One of the top ways, depending on the kind of bacteria you are, that an organism or a bacteria can survive in a dry environment, if it can make an endospore, it will make an endospore. Which two genus of bacteria can make endospores? Clostridium's one. I'm just going to write it up because you guys are going to see these all the time. Clostridium and Bacillus. Bacteria that can make an endospore, you can survive a dry environment. They'll just go into an endospore state. But most bacteria don't have the ability to make endospores. So they still have to survive a dry environment as well. One of the ways they can do that are several different ways. One, they have a glycocalyx capsule. It's an extra outer carbohydrate shell to help them survive longer in a dry environment. Not indefinitely, but longer. The cell wall itself is another barrier so that it's not losing water. And depending on the bacteria species, anything in the Mycobacterium genus, they have that mycolic acid. And although it's an acid, it's really a what? It's really a lipid. And again, it's like coating yourself in a waxy covering. It's going to help decrease dehydration as well. So bacteria are like, okay, well, we can't survive on surfaces because I could sneeze on this table right now and there's probably bacteria on it, but they're not going to survive forever because there's no water, there's no nutrients on the table. But they want to try to survive as long as possible so someone else can touch them and they can find a happy place. So if they have, a cell wall, that, mycolic acid, whether they can make an endospore, whether they have that nice capsule on it, these are all ways for them to help, survive as long as they possibly can in a no water environment. Now, water itself can also pose potential issues when it comes to osmotic pressure. And when I'm talking osmotic pressure, anything osmosis is water. Osmotic pressure is talking water pressure. There are some microbes that require a high water pressure. They're called halophiles. They require a high water pressure. I'm going to go down to hydrostatic pressure. Hydrostatic pressure. What movie is this from? Finding Nemo. Where are they in this particular little picture? Where are they in the ocean? Yeah, they're down in like the deepest, darkest, trenches. And that's bacteria that's in there that makes that glow. There are bacteria that require a high water pressure. At the bottom of the ocean, at the bottom of those trenches, there's so much water, pressure, weight of water pushing in on the cell on every single side. And the microbes that live down there have adapted to it and they require it. If you took this cell, something from the bottom of the trench, and brought it up to the surface where it didn't have that water pressure pushing in on it, that cell would explode. It needs that water pressure to maintain its shape. Now, the other part of water, more osmosis and moving of water, is there are some bacteria that live in a high salt or even a high sugary environment. Halo means salt, so a halophile means these organisms like a high salt environment. Now, if here's my cell, again, I'm not artistic whatsoever, and here's my salt water, like the Great Salt Lake, Salt Lake City, the Dead Sea, which isn't dead, the Black Sea, these all have a ton of salt in it. When you put a cell that has water in it, in a salty environment, where does water want to go? It wants to leave the cell. And in a normal cell, that would shrivel the cell up and the cell will die. But there are some microorganisms that are like, well, we're not going to survive then, because the only place we know to live is in the salt lake. And so they have figured out ways that as water is trying to leave, they can filter the water out of the salt water and water can go back in. But if you put one of these organisms in fresh water, they would die. And so a halophile, these are organisms that require a high salt content. They figured out how to prevent water loss, but make sure water comes in. Now, other than, different physical requirements we have to give organisms, there are some biological associations too, meaning some organisms have to live with other organisms. They can't just grow alone. So a biological association, organisms are living in association with some other different species. Could just be a different bacteria, could be with a whole other organism, like a whole other species. Sometimes they're non-symbiotic relationships, which means they're not always a happy relationship. It doesn't means that they don't always benefit everyone, but they still have to live together. A symbiotic relationship means it's required. They cannot grow without the other organism. And the same, it's not always a happy requirement or a happy situation for all of them, but you'll never find one without the other. There are a lot of parasitic organisms, and pretty much anything that causes harm to us is considered a parasite. But there's a lot of parasitic organisms that you can't find one without the other. They can't survive on their own, but it's not a happy survival for everyone. Now, biofilms is when you have different organisms living together. They don't always have to be the same species, but they're using that glycocalyx, they're using the little fimbriae to attach to each other and attach to surfaces, and they're called biofilms because it literally just means a living covering. So it's a synergistic relationship among numerous microorganisms, and because they all stick together and they cover lots of surfaces, it benefits them. I've got several slides on this. This is showing an example of a biofilm on your teeth. And again, we're going to scrape teeth, I think a week from Monday. And we're going to see what's growing on your teeth. Now, as bacteria land on your teeth, they themselves probably are not going to stick very well. You know, they've got some fimbriae, they've got some glycocalyx that helps them stick, but then you drink something and they might wash right away. So before bacteria can really start to grow, they first have to find a home, and they have to make that home their home. And so they will start to stick to each other, but then as soon as they stick, as fast as they can, they start to secrete a sticky matrix that helps them stick better to the surfaces so that when you take a drink of water, they just don't rinse off. And it also allows more bacteria to stick. So now they almost have their own protective sticky covering. So even if you drink water, they're not coming off. But again, it's beneficial. So benefits to the organism by living in these different biofilms. And again, if something's good for the bacteria, it's always bad to us. It allows them to stick to each other. It allows them to stick to surfaces. By allowing them to all stick together and even having that sticky matrix, sometimes nutrients, like all the food you eat, stick to it. So even at the end of the day, more stuff is more likely to stick to your teeth at the end of the day versus the beginning of the day. That sticky matrix is trying to trap food to feed your bacteria. But it protects those bacteria. And all good things for the bacteria, all bad things for us. Now, our teeth, this becomes an issue because you now have bacteria on your teeth, and the only way you're getting them off is you now have to physically scrub them off, teeth brushing. But you can also kill them when you use mouthwash because that's alcohol-based, and most bacteria can't survive that alcohol. I don't know. They've got something in there. I looked at it the other day because we just bought some, and I'm like, this intrigues me. And I can't remember. There's still a disinfectant that's just not alcohol. I'm like, I just can't remember what it is because I know we have some at home. There's some, because I was like, well, it's got to have something that kills the bacteria. That's the point of mouthwash. It's just not alcohol-based, so I don't think it's quite as strong. Sometimes that's why it has a burning sensation if you've got a cut or scrape in your mouth. It's alcohol-based. Now, other than these biofilms forming on our teeth causing an issue, and we'll go over this later and more times this semester, bacteria on your teeth cause issues. Eventually, they're going to lead to cavities. Why that is, though, because bacteria themselves, you're like, oh, they're just happily sticking on there. The problem is every time you feed a bacteria sugar, bacteria take sugar, and when they break it down, they do it through a process called fermentation, and they produce acid. Acid wears away at the enamel on your teeth. That's cavities. So when you've heard ever since you were a little tiny kid, you know, especially the day after trick-or-treating, you don't eat all that sugar, sugar causes cavities. Indirectly. The sugar feeds the bacteria makes acid from the sugar, and it's the bacteria's acid that forms the cavities. Sugar alone does not cause cavity. I mean, you ate the sugar and then quick brush your teeth, you'd be fine. But no kid ever does that. So ultimately they do, but it's an indirect process. It's those bacteria, the biofilms on your teeth, and you guys probably all want to go home and brush your teeth right now. It's those bacteria on your teeth that are doing it. Sometimes you can even feel this biofilm like at the end of the day, and you're like, oh, ew, it's like icky on my teeth. Yeah, that's biofilm, that's matrix, that's all those things sitting on there. But just because it causes a cavity is not some of the biggest issues. The problem is, as bacteria stick to surfaces, and they start to secrete this matrix, and they start to acquire other bacteria, they can be protected from some chemicals, they can be protected from some disinfectants as they're in their own little protective sticky matrix, which means it's harder to kill them even on medical equipment. So we started the video Monday, and you'll finish it Monday with Leanne. Addie had to go on the ECMO machine. And they even said, like, she started off with MRSA, but that's not really actually what made her sick, like so sick that she needed a lung transplant. As soon as they put her on ECMO, then she picked up a new bacteria, Stenotrophomonas, that was sitting in a biofilm in the tubes of the ECMO machine. You're like, oh my God, how can that happen? It happens all the time. And I'm like, yes, bacteria land on things, And they become almost impossible to kill once they're in their own little protective matrix, and it becomes a huge issue. So we can find biofilms on the ECMO machines. Again, they try to sterilize everything they possibly can, but things happen. Catheters, those catheter tubes, catheter bags, biofilms form on them a lot. It's one of the top reasons why people that have catheters are prone to getting urinary tract infections, is because of the biofilms that's on those tubes, and why they should get changed out as frequently as they possibly can. You gotta scrub them off. So we're trying to figure out ways, because we know different bacteria can land on surfaces. They can land on medical equipment. Some of those bacteria can be, unfortunately for Addie, very resistant to different antibiotics. They can cause lots of issues. So our thing we're doing a lot of research now is, how can we stop biofilm formation from happening? How can we prevent bacteria from sticking to surfaces? How can we prevent them from sticking to each other? If we can stop them from forming biofilms and sticking, we can prevent a ton of diseases. We can also stop a lot of cavities too. There's a new toothpaste. I haven't seen it hit the market yet though. It's been approved, but in the United States, everything takes forever. So I think you can buy it in Europe. That actually is preventing biofilm formation in the toothpaste. So there's something in it, I don't remember what it is. I read the article on it, that's preventing it. So we're getting there. We're doing research on it. Now, our next little unit is, how do we grow organisms? So we know what they need. So how do we grow them in a lab? Because that's what we're going to do this semester, is we're culturing organism, which means we are helping these organisms grow and divide. So how do we grow microorganisms? Well, an inoculum, that's the term for an initial sample, is introduced into a medium, which is going to be some type of growth nutrient factor, whether it's a fluid, whether it's an agar. So we're going to take some initial sample and we're going to put it into some nutrient-enriched media. Now, where that initial inoculum specimen comes from is one of three sources: an environmental, clinical, and stored. An environmental sample is if I wanted to go out and take a water sample from the Mississippi and find out what's growing in it. I don't want to know. Ignorance is bliss. But I could. And that's where my initial sample comes from. I can find out what kind of bacteria is in soil. I can find out what kind of bacteria is in the air. Those are all environmental specimens. In our class, we don't care about those as much. Clinical specimens means the initial sample came from a patient, which is what you guys are all going to be working with. Initial samples are coming from the patients, and it's usually to identify and diagnose a patient. Stored specimens are ones that are usually used for research. So they're grown in a lab, they're multiplied in a lab, they're stored in a lab. So when Arlene pulls out samples, or Leanne brought some in from her husband Bill, 'cause I guess if you work here, you marry a microbiologist too. We both married microbiologists. When we want to work with bacteria, so on Monday, you guys are going to get tubes of either E. coli or Staphylococcus epidermidis. Those samples didn't come from a patient, and they didn't come from an environmental source. We pulled them out of our -80 freezer, and then we got them going. And so when people do a lot of research, they're not going out and like, well, let's just see if I can find a patient that's got the particular bacteria. We can have stored specimens of different bacteria as well. I mentioned before, culturing is just the act of cultivating or growing bacteria. What we're looking for, and sometimes counting on the plates, are colonies, knowing that a colony is not just one bacteria. It's where one bacteria landed and multiplied. So the more official definition of a colony is called a colony-forming unit, because everything in that colony originated from one bacteria. One bacteria landed, it became 2, then it became 4, then 8, then 16, then 32, and so on until it becomes 5 million bacteria, visible as a colony. So as one bacteria landed and constantly reproduced, but I could visually see it and count it. So even though I can say, all right, well, there's 50 colonies right here, it all originated from 50 single bacteria. Now, anytime we're growing bacteria, we always want to try to do it aseptically. So aseptic technique means reducing the chance of contamination. And on Monday, Leanne will go, worked out perfect that she stopped in. Leanne will go over a little bit more ways that we try to do everything aseptically, as we're now going to start growing bacteria. And when we give you a tube E. coli or Staphylococcus bacteria on Monday, hopefully that's the only thing that's growing on your plate. And so different ways we already do aseptic technique is that we wear our PPE so that we're not contaminating things. We spray down our table at the beginning, we spray down our table at the end. You'll be using incinerators on Monday. We try to do everything aseptically. We try not to open the plates up unless we're actually doing something with it. It's all things to try to reduce contamination. Oh, I don't know why my little video didn't work. Oh well. Now, when we're working with a pure culture, that means all the bacteria are one species, and next week that's what you're going to get. You're either going to get E. coli or you're going to get a staph bacteria. But you're going to get a broth culture. It'll be a fluid, and it's going to have, I don't even want to estimate how many bacteria, billions of bacteria in there, but we want to decrease the number of bacteria on the plate so that we start to get individual little colonies so that we can see what is their shape, what is their size, what is their color, all of that, so we can try to identify it. So we're going to do something known as a streak plate. This is what you're going to do on Monday with Leanne. She's going to show you how to do the streak plate method to try to go from where you have lots of bacteria to a very small amount of bacteria to get isolated colonies. This is showing a mixed culture. You can see that there's multiple bacteria species together. And when you get them really crowded, you don't even notice in these first sections where there's so many bacteria that there's actually 3 different bacteria. You really only see 2. But when you start to isolate them enough and they can all grow happily, then we can start to see their size. You notice they start to get bigger as well, but we can start to actually see different bacteria. You guys are going to get one kind of bacteria, and some of your plates might look just like this. But the streak plate method, you're taking an area on the plate where you put lots of bacteria, where you don't expect any kind of isolated colonies. And through using your little loop on Monday, you're going to pull some of those large, just a small amount from that large area onto another section, and then some of that to another section, some of that to another section, constantly decreasing the number of bacteria. So that hopefully you get some isolated colonies, but either kind of the 3rd or the 4th sections on your plate. That's one way we can get isolated colonies when we have a really high concentration. It's probably the most common way, but it's not the only way. Another way is called the pour plate method, is if I give you a broth that has billions of bacteria into it, if you just poured some of that broth on the plate, it would be too numerous to count. You'd never see individual bacteria. But you could start to dilute the broth and start taking just a little bit of that liquid and put it into a sterile tube. And you could take a little bit of that and put it into another sterile media tube. And you could dilute some of that and put it into another, and then pour some of that on the plate. So then you don't need the loops at all, but you can dilute the broth enough. The problem is you don't always know how far you have to go with dilutions. And that requires a lot of tubes. And so that's a lot of media. You can reuse tubes and autoclave them, but that's a lot of media, and potentially that's a lot of plates because you don't know which plate, which dilution is going to give you isolated colonies. So streak plate method, more common, less supplies. Culturing media, again, we do have to give the bacteria different types of media to grow on. A lot of the media, not all, but a lot of the media has some type of agar to it, which is a thickening agent. It's what gives it that consistency of Jell-O. Now, it's different. That's gelatin. This is agar. Agar originates from a red algae. And they added to it because bacteria can't use agar for anything. It's not a nutrient, so it's not meant to grow or inhibit bacteria. It's just meant to thicken it. And there are, as my picture shows, lots of different types of plates and media. Lots of different combinations of nutrients, lots of different combinations of growth factors. Sometimes they're plates, sometimes they're tubes. We're going to use a lot of different types of things this semester as well. But even though we have lots of different types of media to try to make every bacteria as happy as we possibly can, Most prokaryotic organisms out there, we still have yet to be able to figure out what media can we give them that we can grow them in a lab. We figured out a lot of them, but we haven't figured them all out yet. Some of the things that we'll use this semester other than plates, because we're going to use blood agar plates, you'll use those again on Monday. One of the things that can be commonly used is known as a slant tube. So we pour sterile agar in it, but before we allow it to harden, just like Jell-O when you allow it to harden when it cools, we tip the tube just a little bit, and it provides 2 environments in one tube. We don't know what patients have. That's the idea, is we're diagnosing it. And we don't know if whatever they have is aerobic or anaerobic. A slant tube is really great for growing bacteria when you don't know if they're aerobic or anaerobic, because that slant region gives them a huge aerobic area to grow, and then the butt region still gives them a area to grow anaerobically. We will use these later this semester, but we will use them. Now, some terms, and this is my last slide. I was trying to remember, I recorded a lot of stuff yesterday. I'm like, I can't remember, I don't think I recorded this. I think I started right here. Some terms for general culture media. A defined media, I'm not writing the word up, it's up there, means the exact chemical makeup is known. Meaning, I know exactly what's in that media. And you're like, well, shouldn't you always know what's in that media? Not always. The more common one, because this is actually not even that common, even though when we make media, we measure everything out and add it to it, depending on what we're adding, some of the stuff, as soon as you add it into the media, may start breaking down. And you don't know in what percentage it's broken down, so then you don't know the exact chemical makeup. So complex media, I don't know how I want to define this. The makeup, we'll say it's partially known. I'm going to give you some examples. We can add into our media milk, and we know that will break down, but there's a lot of bacteria that need the nutrients in milk. We can add in beef. Beef extract is a super common nutrient added to a lot of media. We also know it breaks down when we start adding it to it, but we know all the nutrients are there, but we know it breaks down, so we don't know the exact makeup of it. Soy is another protein we like to add, and we know that starts to break down into its amino acids. The blood in a blood agar can also break down. All the nutrients are still there. We just starts to break down. I think even yeast is another one we know will start to kind of break down. It's very common to have a complex media versus a defined media. There's lots of nutrients we can add. We know the organisms need everything that's in the nutrients. We know they need everything in the nutrients even as it's breaking down, but as it's breaking down, we just don't know the exact formula for what's broken down and what isn't. Selective media, we're going to use in our lab this semester. Does this go to 1050, right? 1055? Okay. I was like, right, it's not 1040. Selective media, because you're going to see me define it. It'll be in lab again. Let me see. There is, I'm trying to think of a way to write it. Because I like to write it as short as I can. Chemical, I don't know if that's the right word, is added that inhibits one type or group of bacteria, but has no effect on another type or group. And I will give you an example in a second. That word is group. I think I recorded part of this. So we will use selective media in the lab. I recorded this so you can skip ahead when you watch some of this. This is showing an example of a selective media versus a nutrient media. And some of you guys actually saw examples of this if you swapped the bottom of your shoes this last week and looked at it on Monday. So nutrient augers meant to grow anything and everything. It's a nice neutral pH. It's got tons and tons of nutrients in it. Bacteria grow crazy. But the one thing, when you've got lots of things all growing together and the bacteria that are going crazy, everything's competing for space and nutrients. And bacteria will always outcompete fungus. So if we want to know, well, does a patient have a fungal sample? Do they have a fungal growth on their skin? If I did a swab of your skin, I'm probably only going to see a bunch of Staphylococcus bacteria from your skin, and I want to know, does the patient have fungus growing on their skin? We use media to inhibit the growth of bacteria. So there's something in there, and in this example, it's acid. An acid is added that's going to inhibit one type or one group of organisms. For us, it's just going to inhibit generally all bacteria. But then it has no effect on what we do want to grow, in this case, fungus. So it's a selective media because it's selecting for one type or one group of organism. So there's something in it that's going to inhibit everything else except for what we want to see growing. Differential media, there we go. There is a substrate added to determine if bacteria can do something with it. How we will know this? Color change. We can add stuff into our media. We can add substrates. Sometimes it's different sugars. Sometimes it's different proteins. And we're just asking the bacteria, can you do something with it? And if it can, it produces some product, and we add something in there to make sure that a color change will happen. We're going to use differential media in our lab as well. I think it's maybe like 2-3 weeks from now. I'm just going to use examples again. Our blood augers are actually a differential media. The red blood cells are a substrate because some bacteria can break down red blood cells fully. Some of you saw that on your plates on Monday, that you could look right through the media. Some bacteria fully broke down that particular substrate, the red blood cells. Some bacteria just break it down a little bit. You can't see through it, but it's not red anymore. It's more of a greenish color. And then some can't at all. I know this is recorded. This is showing, this is a plate that we're going to use in a couple weeks called the macaque. Over on the far side here, we've got two different types of bacteria. They're both gram-negative bacteria. They will happily grow on this particular plate, but they're different colors. Even though they're both a gram-negative bacteria, to help us identify which bacteria it is, we can look at which color change happened. And on this particular plate, the color change we're looking for is the bacteria show up as pink. And so we're like, oh. And E. coli on a macaque plate will show up as pink bacteria. It helps us identify bacteria as well based on if color changes happen. And then my last, and I think I actually have a separate slide later for this. Transport media is meant to keep bacteria alive, and that's it. Not grow. And I know I've got a slide recorded on this. The idea is when you go to the doctor and they collect some type of specimen from you, throat culture, nose culture, sputum sample, I don't know, urine culture, it usually will go into some type of transport media. which means there's a minimal amount of nutrients in there so that if there are bacteria present, the bacteria stay alive until they can get to the lab and the people that work on the lab can analyze it. It's not meant to grow bacteria. It's just meant to keep them alive for a short amount of time. Because if you have 10 bacteria at the back of your throat, you want to keep all 10 bacteria alive so that the people in the lab can identify them. You don't want those 10 bacteria to become 10 million bacteria and they think everyone has raging infections. But they also don't want those 10 bacteria to die and then not diagnose it. And so transport media is meant to just keep organisms alive for a short amount of period or amount of time, and that's it. That's where I'm going to end. Let me stop this.